Hello to everyone here who are participants. Today we will be talking about probing the biology of cell migration and invasion using phenotypic live cell imaging. My name is Tim O'Callaghan and I'm an application specialist at EsenBioScience. So before moving into specific information around cell migration and invasion, I would just like to give a brief introduction to our approach to phenotypic live cell imaging. We call this approach live content imaging, and it draws on a number of factors from many different technologies, such as the automated image analysis and quantitative plate-based pharmacology associated with high content imaging, and also the ease of accessibility and the high quality phase contrast images of light microscopy. These are combined to provide real-time resolution of spatio-temporal phenotypic changes, and this is provided with simplified plate reader interface and intuitive analysis tools. So live content imaging is defined as the acquisition, analysis, and quantification of images from living cells. These are cells that have not been fixed and stained, that remain unperturbed by the detection method, whether that's the imaging process itself or the use of reagents used to identify phenotypic changes, that allow for repeated measures over longer periods of time, from days to weeks as opposed to minutes to seconds. We achieve this using our live content imaging device called InvasionMigration Zoom. And InvasionMigration Zoom is a fully automatic two-color phase contrast and fluorescence microscope designed to fit inside a standard tissue culture incubator. It can accommodate a wide range of standard cell culture vessels, and it is ideal for kinetic long-term live content imaging. So cell migration is defined as the movement of individual cells, sheets, or clusters of cells from one location to another. It plays important roles in many physiological and pathophysiological processes, including development, such as embryonic morphogenesis and angiogenesis, regeneration, including wound healing and atherosclerotic lesion repair, and also diseases such as metastases, which are characterized by the uncontrolled movement of cancer cells away from the site of the primary tumor and to distant sites within the body to establish secondary tumors. In contrast, diabetic ulcers are characterized by the insufficient migration of cells into the wound area, resulting in a failure to epithelialize the wound and failure for wound closure. Throughout this presentation, I will be referring to cell migration as the movement of cells across a substrate, and cell invasion as the movement of cells as they penetrate through a three-dimensional extracellular matrix. And it's cell invasion that is typically associated with cancer cell metastases. The cell player kinetic migration and invasion assays are an extension of the scratch wound model. They employ a 96-well wound maker and image lock plates to create consistent wounds in all wells of the 96-well plate. The plate is then placed within InvasionMigration Zoom, and images are acquired over the course of the assay. Migration is quantified as cells move into the wounded zone, and the resulting metrics can be used to plot time courses and generate kinetic pharmacology. The assay is simple, reproducible, and kinetic. The 96-well wound maker is a precision-engineered device, and it is designed to create wounds in each well of the 96-well plate in a single operation. It is fitted with 96 steel pins, each tipped with PTFE, polished PTFE inserts that are calibrated to apply uniform pressure to each well of the plate. This is so that we do not remove any underlying biomatrix coating if present, and also so we do not damage the border regions of cells following wounding. The wounded plates are placed within InvasionMigration Zoom and imaged at regular intervals. for example, every 2 hours or 3 hours over the course of the assay. The user has a choice of 3 objectives to acquire the images, a 4x, a 10x, or a 20x. And the images can be concatenated into movies of cell migration or invasion. And this is a movie illustrating HD1080 migration over plastic. And if I play it, you should be able to see cells moving uniformly into the wound area. Now, if you're having trouble visualizing this movie, I would encourage you to move to the drop-down menu around here and open up the menu and select the 720p resolution, and that will optimize your performance for you. So these movies can be used to document cell morphology and behavior over the course of the assay and in the presence of different treatments. The cell migration protocol can be performed either with uncoated or wells coated with a thin layer of extracellular matrix. Cells are then seeded on top of this substrate, and once they've grown to confluence, the 96-well wound-making tool can be used to create wounds in each well of the plate. After wounding, media containing treatment is overlaid on the wounded monolayer, and the plate is simply placed within InvasionMigration and images are acquired. For cell invasion, it's essentially the same process. However, this does require a well coating of the extracellular matrix of choice. Cells are seeded on that extracellular matrix and at confluence wounded, and after wounding, the extracellular, a 3-dimensional extracellular matrix containing drug treatment is added on top of that wounded monolayer. After the gel has had time to set, we apply media, again containing compound, on top of the gel, and the plate is then placed within InvasionMigration for imaging and quantification. At Essen, we've validated both collagen 1 and Matrigel for use in this assay. Images can be analyzed using the integrated image analysis metrics, and these are user trainable and can be used to optimally mask your wound and cell movement into the wound over time. So the left-hand image here, immediately after wounding at T0 hours, shows the initial scratch wound mask in green. As cells migrate into the wounded area, they are illustrated with the blue mask, and this is continued until full wound closure is observed. 3 metrics are reportable from these analyses, the first being wound width, which provides the mean distance between the wound borders over the course of the assay. It's a metric that starts off at a high value, around 720 microns, and falls to a lower value as the borders move towards each other and ultimately the wound heals. The wound confluence metric reports the cell confluence within the wound area over time. This metric starts at a low value and increases as cells repopulate the wound area. The final metric, relative wound density, is a very robust metric, and this reports the cell density within the wound region relative to the non-wound region. It is a metric that starts at 0% at the start of the assay and tends to 100% following full wound closure. A value of 20% would correspond to a wound density, a cell density within the wound of 20% that of the non-wounded area. It's a self-normalizing metric and can account for changes in morphology, which may be caused by treatment effects, and also changes in cell density outside of the wound that may have occurred due to cell proliferation over the course of the assay. The migration time course is highly consistent across the plate, And this plot shows the relative wound density metric values for all 96 wells of a HT1080 wounded plate over time. And we can see here that there is a very tight distribution of relative wound density values for each well, such that the variability across the plate is very little. The histogram insert is a histogram of the average wound widths at time 0, and we can see that they center around 730 microns and have a coefficient of variance of under 2% in this assay. The assay is compatible with many different cell types, ranging from primary cells to immortalized cells, cancer cells, and stem cells. And it has been shown to be compatible with over 40 different cell lines, and this is from data generated in-house and from external labs that have published data using this assay. Just about the only cell type that is not compatible with this assay are suspension cells and non-adherent cells. On the left-hand side, we've got a plot of multiple migration time courses for cancer cell types in blue and also non-cancer cell types in the open symbols. We can see here that the HT1080 fibrosarcoma cell line is very quick to repopulate the wound, and full wound closure is observed after around 12 hours. This is a markedly different profile from the rat myoblasts H9C2 cell line, which doesn't fully repopulate the wound until around 50 hours. IncuSight Zoom provides a very nice microplate view of the temporal time courses for the experiment. And this is really helpful to allow you to compare treatment effects across the plate. In this experiment, HT1080 cells were plated in each well of the 96 well plate, and they were treated with increasing amounts of cytochalasin D. And as the concentration of cytochalasin D increases, we can see a decrease in the rate of wound closure for the HT1080 cells. If we average the replicate rates of wound closure and draw them on a single axis, we can more clearly define the differences in migration rates under these different treatment conditions. Vehicle control rate of migration is shown with the white open symbols, and as we increase the concentration of cytochalasin D, we see a decrease in the rate of migration. And for cytochalasin D, which is an actin polymerization inhibitor, we can see that this inhibitory effect is largely time independent. This is not the case for other pathway inhibitors that we've tested. For example, the HSP90 inhibitor CCT018159 shows very little early phase inhibition of migration. However, towards the later stages of the assay, we see a marked inhibition, especially at the top 2 concentrations, with actually ablate wound closure. And so we only see partial wound closure here for these concentrations. This profile can be contrasted with the profile for the PI3 kinase inhibitor wartmenin, which shows an early stage inhibitory effect that subsequently recovers and allows full wound closure at the later time points. And these two profiles really highlight the fact that it's very difficult, not impossible, to choose a single endpoint measurement that captures the degree of temporal variability that is seen with these compounds. The top right-hand panel shows the mTOR inhibitor KU00637994, and this has an alternative kinetic profile that shows mild to modest inhibition over the whole time course of the assay. Now, with an endpoint analysis for this compound, it might be very difficult to be convinced that there is true biological differences between the treatment effects at the single time point. However, for a kinetic analysis, we can provide extra confidence due to the repeated measurements. And these can be used to provide confidence that the differences, the observed differences, are biologically significant. So these 4 treatment profiles really show the value of kinetics and show that kinetic analyses are really important to fully understand the temporal effects of these compounds. Now we can analyze these temporal profiles further by creating compound signatures for these 4 compounds. And we did this by creating concentration response curves for each time point of the time course. And from these concentration response curves, we generated measurements of potency using a PIC50 measurement, and also a maximal inhibition for each time point. And for cytokinase D, we can see that potency measurement decreases with time. However, the maximal inhibition observed at each time point is constant at 100%, illustrating that complete inhibition of migration is observed at all time points. We can now compare this temporal pharmacological profile with those for the other compounds, and for the HSP90 inhibitor, we see that at the early phases we saw very little inhibition that increased over the time course of the assay and ultimately prevented wound closure at the maximal test concentrations. In contrast, Wortmannin showed early time point inhibition that decreased as cell recovery occurred over time and ultimately allowed full wound closure towards the later stages of the assay. The mTOR inhibitor showed a low level of maximal inhibition over the whole time course. And this really shows that these temporal analyses allow us to identify these low levels of inhibition in a robust manner, even at early time points. So behind each of these temporal profiles and analyses are also the images and movies that can be used to support conclusions drawn from them. So if I now move over to the movie for these compounds, we can compare the morphological effects caused by these compounds over time. In the control wells, we can see that HT1080s migrate to close the wound in a very uniform fashion. This is strikingly different from the phenotype observed for the HSP90 inhibitor, which shows very minimal early phase inhibition, but very marked late phase inhibition. And the cells really stop in their tracks as they're migrating to close the wound, and ultimately wound closure fails to occur. The Wortmannin PI3 kinase inhibitor shows early phase inhibition, so the cells don't really move from the wound border regions over the early phases, but then recover and move into the wound area. And the mTOR inhibitor shows very marginal inhibition over the time course as compared to the control. However, closer phenotypic analysis of the images show that the cells may be more hypermotile in the presence of this mTOR inhibitor, and also the directionality of migration may be lost somewhat, and perhaps these phenotypes are associated with the low level of inhibition observed for this compound. Of course, further phenotypic analysis would need to be performed to determine this, but it's an interesting observation nonetheless. We can also investigate cell invasion using this assay format. This is performed by overlaying the wounded monolayer with an extracellular matrix and observing the cell penetration through this matrix over time. Cell invasion is characterized by a very different morphology as compared to migration, and also a much slower time course. So if we look at the right-hand video of HT1080 cell invasion through Matrigel, we can see that the cells display a mesenchymal-like morphology and extend spike-like invader podia into the Matrigel as it's remodeling the extracellular matrix and moving in to close the wound. The time course, of course, is also very much slower than that of the HT1080 migration. Further validation of invasion was performed by showing that non-invasive cell types do not invade the extracellular matrix. We performed an experiment using two highly invasive cell lines, the HT1080 and MDA-MB231 cells, in combination with a non-invasive MCF7 cell line. Under control migration conditions, we can see that full wound closure was observed using for all of the cell lines. However, only the invasive HT1080 and MDA-MB231 cell lines could penetrate the extracellular matrix and begin to close the wound. The MCF7 cells remained non-invasive over the whole course of the assay. An assay such as this could be used to determine the mesenchymal to epithelial to mesenchymal transition of cells within an assay format. This slide really illustrates the difference in invasion and migration kinetics. These highlight that invasion through a collagen 1 gel is much slower than the rate of migration over a collagen 1 coating. And for HT1080 cells, we can see that migration in the open symbols is complete by roughly 12 hours, whereas the rate of invasion is much slower and wound closure doesn't complete until around 50 hours. The same trend is shown for the MDA-MB231 cells, and the MCF7 cells can only migrate and do not invade through the collagen 1 gel. Pharmacological differences are also seen between the invasion and migration assays, and in this HT1080 invasion assay through collagen 1 matrix, we have tested a broad spectrum matrix metalloprotease inhibitor GM6001 and shown that that exerts preferential inhibition of invasion through collagen and no effect on migration of the cells over a collagen 1 matrix. Similar effect is seen for the myosin 2 inhibitor blebistatin, which showed a marked degree of concentration-dependent inhibition of invasion and only a modest effect on the cell migration time course. We can also probe the effect that the extracellular matrix has on the cells themselves by running an analogous assay, but this time using Matrigel extracellular matrix. And under these conditions, the HT1080 cells were not affected by the presence of the MMP inhibitor GM6001 for invasion, and likewise they showed no effect for their control migration modes. In contrast, however, we saw for the myosin 2 inhibitor blebistatin that invasion was inhibited for the HT1080 cells through BD Matrigel, and again only a modest inhibitory effect was observed for migration. Users can also use these assays to investigate migration of invasion phenotypes in mixed cultures. In this experiment, we pre-labeled non-invasive cell type MCF7s with a green fluorescent protein and also a HT1080 invasive cell line with a red fluorescent protein. And from the phase contrast images, we can see that it's very hard to discern the different behaviors that each cell type shows over the course of the assay. However, in the fluorescence channels, we can see quite clearly that only the red HT1080 cells move into the extracellular matrix to repopulate the wound, and the MCF7 cells remain non-invasive over the course of the assay. So in summary, the cell migration and invasion assays are fully kinetic, label-free 96-well assays with integrated analysis tools that allow quantification of cell movement across the substrate or through an extracellular matrix. They provide morphological information through images and videos and support detailed pharmacological analysis and phenotypic screening. The 96-well wound maker enables simple and reproducible wounding and has been shown to work with many different cell types, including challenging low contrast cell types such as HUVEC. The kinetic data allows retrospective identification of the optimal time point for measuring drug effects, and also enables full time course analyses such as area under the curve analyses for pharmacological profiling. So I hope that I've convinced you that detailed phenotypic analyses add value, and that kinetics and morphology are important when evaluating treatment effects, as they provide a better understanding of the behavior of cells within a system and are critical when evaluating the temporal effects of a treatment. I'd now like to move on and show how independent labs are using these assays in models of epithelial to mesenchymal transition, and also neurogenesis, and in models of angiogenesis. So this slide illustrates 3 independent studies performed by independent labs for pathway analysis in tumor cells, illustrating combination pharmacology, gene overexpression, and shRNA knockdown. The left-hand research was performed by the University of Helsinki, which showed that salinomycin reduced PC3 prostate cancer cell migration, and that this effect is antagonized by the co-application of the antioxidant vitamin C. So these data we used to support the conclusion that the effects of salinomycin were caused by oxidative stress induced in the PC3 cells. Central panel is research conducted at Genentech, and they investigated the role of adiponectin receptor 1 in breast cancer cell invasion. Adiponectin receptor 1 is often underexpressed in aggressive breast cancer cell types, and they overexpressed adiponectin receptor 1 in a basal-like cell cancer type, MDA-MB231 cells, and they showed that overexpression actually reduced the rate of invasion of breast cancer cells into a matrigel matrix, and also attenuated epithelial to mesenchymal transition. The team also used IncuCyte to monitor the growth effects of overexpression and showed that the overexpressing cell types showed no significant difference in growth compared to the parental controls. A team at University College London showed that downregulation of the autophagy regulator DRAM1 impaired migration of glioblastoma stem cells, and the team highlighted that DRAM1 could provide a new target for drug therapy against aggressive glioblastoma multiforme brain tumors. Independent labs have also probed the biology of vascular endothelial cells and how that relates to the angiogenesis and atherosclerosis biological processes. Napoleon Ferrara's lab at the University of California, in combination with Genentech, showed with a HUVEC migration assay that a HUVEC migration screen they actually screened VEGF phosphoproteome siRNAs for effect on HUVEC cell migration. And in the study, they identified fork-head box protein 01 as a transcription factor that inhibits VEGF signaling. They actually went further and used EsenBioScience's co-culture model of angiogenesis to further validate VEGF signaling mediators of angiogenesis. The central panel here from research from the University of Connecticut showed that simvastatin, a statin-like compound, increases endothelial cell migration compared to a control. And this observation was consistent with the pro-angiogenic effect of statins on the ischemic heart diabetic rats following myocardial infarction. And the team suggests that this pro-angiogenic effect is primarily associated with the pro-migratory effect attributed by simvastatin treatment. And finally, on the right-hand side, Fearon et al. At British American Tobacco created a HUVEC model of smoking-induced atherosclerosis. And they showed that HUVEC migration was inhibited by cigarette smoke particulate matter. And inhibition was reduced when cells were exposed to particulate matter from an experimental cigarette with reduced smoke toxicant levels. And these data are shown in the bottom panel here, with the experimental cigarette in the dark symbols and the reference cigarettes in the open symbols. And on the left-hand side, we've got measurement of wound width, and we can see that wound closure is fastest for the experimental cigarette with reduced smoke toxicant levels. Uden et al. At King's College London probed the biology of neurogenesis in the adult using a neuronal stem cell migration model. So neuroblasts migrating in the adult brain can be redirected by cannabinoid signaling to sites of brain damage, and this redirection may be associated with brain regeneration. The team at King's College London investigated neural stem cell migration in the presence of endocannabinoid inhibitors, and they showed that inhibition of cannabinoid receptor 1 using compound AM251 actually inhibited the rate of migration compared to control. In addition, the application of cannabinoid receptor antagonist, the cannabinoid receptor 2 antagonist JTE907 compound also inhibited neuronal stem cell migration, and co-application of these two antagonists further inhibited migration. Further, they showed that inhibition of diacylglycerol lipase, the enzyme that creates a primary cannabinoid molecule in the brain, also phenocopies this inhibitory effect in a concentration-dependent manner. And here they use an RHC antagonist to DAGL. The team also probed activation of the cannabinoid receptors, and they showed that activation of cannabinoid receptor 1 using cannabinoid agonist for receptor 1 actually increased the rate of migration compared to control. And this increase in migration could be attenuated by the application of the cannabinoid receptor 1 antagonist, and these effects are reproduced using cannabinoid 2 receptor agonist and antagonist pairs. And finally, IncuSite images and movies can also be exported to allow bespoke analyses in third-party software such as ImageJ. Command et al. At the Institut Pasteur in France used astrocytes and U37 astrocytoma cells, and these cell monolayers were wounded and cell movement was recorded in InvasionMigration. Individual cells were tracked by analyzing the InvasionMigration movies using ImageJ software, and the diagrams on the bottom right here show the movement of those cells, or 10 representative cells, over the course of 36 hours. The team showed that downregulation of end-coherent levels in glioblastoma cell lines correlates with non-directed migration as compared to the control astrocytes. They also showed that loss of end-coherent in primary astrocytes and glioma cells leads to a faster and less directed migration, and that exogenous expression of end-coherent reduces glioma cell velocity and begins to phenocopy the control cell astrocyte cell movement pattern. So in summary, there have been over 45 independent publications using InvasionMigration migration and invasion data. And these publications are growing on a weekly basis, and they follow the same trend as the number of publications about our InvasionMigration technology as a whole. And on the right-hand side, I've got a bar chart showing the increase in InvasionMigration publications since 2007 to date. And to date, we have over 185 publications that have used InvasionMigration live content imaging to support the conclusions in the paper. So we and others have used live content imaging to probe the biology of migration and invasion, and to determine treatment effects on phenotype, paying particular attention to kinetics and morphology. The assays are compatible with small molecules, RNAi, and gene overexpression, and they can be used to assess combination treatment effects also. Independent labs have used these assays to screen for functional effects on migration and to develop models of metastatic potential, epithelial to mesenchymal transition, and models of tissue regeneration. Labs have also compared the migratory profiles of different cells and also been able to compare the migratory profiles of isogenic cell pairs. In addition, movies and images can be exported to allow bespoke analyses in third-party software to enable you to develop analyses that suit your needs. So really, I hope that I've given a good overview of our high content imaging, cellular content imaging device, and hopefully provided you with some incentive to delve into the literature and find out what independent labs are using our technology for. So with that, I thank you for your attention and open the floor to questions. So I'm now moving over to look at questions that have been asked during the course of the webinar, and I will just read them and answer them. Okay, so I've got a good question here. Are phenotypic measurements made in 2D space only? Can measurements of invasion be done in 3D? In other words, can you measure invasion in the Z-axis? So that's a fantastic question. And our invasion assays are three-dimensional assays in that the cells have the ability to penetrate the extracellular matrix and move as they wish within the 3 dimensions. However, we can only image within a single Z plane, so we can't perform Z stacks. So in that respect, the measurements are made within a 2-dimensional cell plane. But we feel that that is a very good measurement of cell invasion. Okay, so the second question is, okay, so if cells doubling time is overlapping with migration time, how do we tell that wound closure is due to migration or proliferation? So that's a very good question, and a number of labs use compounds to inhibit proliferation over the course of the assay to prevent this. We don't recommend doing this, and actually we've developed our relative wound density to take this into account. So firstly, let me just state that for migration at least, typically the assays are certainly for fast migrating cell types as HT1080s, the wound closure is complete within, say, 12 to 15 hours, and that's typically faster than the doubling time that would have a significant effect on wound closure. But we've also got our relative wound density metric that actually takes into account changes in cell density over the course of the assay. And if proliferation occurs, we will take that into account with this metric, and it will essentially be normalized out. There are one or two caveats to that, but it's certainly something that we're aware of, and we would recommend the use of relative wound density for that reason. We've got one or two more questions here, so thanks for continuing to ask questions. Is there a way to add chemotactic gradients to the wells? That's a fantastic question, and as with any scratch wound model of migration, this is something that we're not able to do in this model of migration. Of course, we're very aware of the need to understand chemotaxis and being able to resolve chemotaxis from chemokinesis and also haptotaxis, for example. So these are things that we're very, very aware of, but we are unable to address with this model of migration at the moment. So I hope that's answered your questions, but please keep in contact, and that's a wonderful question to ask. A further question is, do we use serum in most of the migration assays? So we do routinely use complete culture media which contains serum, unless required otherwise. Serum starving can be performed if you want to, of course, inhibit cell proliferation. So that's another way to take into account proliferation over the time course. These are techniques that can be performed and are suitable and compatible with this technology. So that's not a problem at all. You can either serum starve or run with full serum as you wish.
Thank you so much for joining us today. Be sure to connect with us on Facebook and LinkedIn if you haven't already. It's a great way to keep up to date with all of our news and special announcements and special content that you can't find anywhere else. So Facebook, Twitter, and LinkedIn, please connect with us. We'll look forward to connecting with you there.